Good morning, everybody. My name is Sonja Guil. My lab is based in Barcelona, in Spain, and I'm going to give you today a brief overview of how human cellular models have been used over the last decade or so to study Rett syndrome. In addition to the wealth of information that mouse models have provided and are still providing in the research of Rett syndrome, the advances in stem cell biology have allowed the development of human cellular systems to model the disease. The idea is to be able to grow in a culture dish in the lab human neural progenitor cells that can then be differentiated into the different neuronal and glial subpopulations that build up the brain. There are different sources of these neural progenitor cells, but I'm going to mention only the one most widely used, and that is tissue from the patient, generally from a small skin biopsy, which is converted in the lab through a reprogramming process into cells capable of now generating virtually every cell type in the body. These are called induced pluripotent stem cells, or iPS cells, and they can be driven towards neural progenitor cells and then into mature neurons. This is a most valuable system, since we can imagine that those neurons generated in vitro are similar to those in the person's brain and with the same genetic background of the patient, including the mutation on MECP2 that the Guil is carrying. Therefore, these cells in vitro constitute a valuable platform to study mechanistic aspects of the disease, as well as to search for biomarkers, or even to use some therapeutic compounds, or even to test gene therapy approaches. The obtention and generation of patient-derived iPS cells, and then from the obtention of neural progenitor cells and mature neuron is a lengthy process and very sophisticated procedure, which with time has been now improved and standardized. The end point is the obtention of these mature red neurons, which generally show most of the defects that had been also observed in animal models or postmortem samples. Specifically, defects in the electrical properties of the neurons. And here you can see some excitatory currents are shown. Consequence of alterations in the morphology and function of these cells. For example, see here in these red neurons the decrease in the green signal, which reflects or detects the synaptic points where synaptic transmission is occurring. And indeed, these human neurons generated in vitro display a number of alterations at the molecular, morphological, and functional level. To summarize, these cells show a poorer morphological complexity, a poorer arborization, a reduced number of spines where synapses take place, and thus altered electrophysiological properties. All these features are characteristic of the disease and also in a full organism. These cellular models are therefore useful to better understand Rett and have taken advantage of the general advances in the field of stem cells. In this arrow of time, I've highlighted some of the most relevant achievements in this area, including the obtention of the first patient-derived iPS cell line in 2009, and then more recently, the obtention of neuronal cultures that are just not growing just on a petri dish on the surface in what we call two-dimensional or 2D neuronal culture, but that are cultured in three-dimensional structures, 3D, called brain organoids, that resembles much more the complex network of cells in our brains. Even brain region-specific organoids can be obtained, and they can also be fused together to study their interrelations. The first obtention of iPS cells from a Rett patient was described in 2010 and was followed by the establishment of protocols to prepare good isogenic controls, meaning that we can now obtain cells from the same patient that one cell line will be expressing the normal MECP2 and the other one will be expressing or using the mutated version. So these are like perfect controls for study. In the following years, a number of labs have focused on the characterization of these cells in 2D cultures. And from 2016, 3D cultures were developed. And soon after, red organoids have been reported. More recently, some studies have achieved diffusion of different brain-specific organoids corresponding to different brain regions, and it is expected that soon the vascularization and also the incorporation of blood-brain barrier mimics will be implemented in this red cellular model. So altogether, the idea is to achieve the most realistic physiological environment. So let's look at some of the examples, of the works that show how important these systems, how important information these systems provide. Here I'm showing some results from the first study in red iPS-derived neurons, showing reduced neuronal maturation, reduced calcium signaling, and impaired firing activity, all indicative of functional defects. Let me highlight here the fact that in this study, treatment with IGF-1, which, as you know, well, trofinetide is an analog of IGF-1, here too showed the ability to recover some of these defects. To the cultures of neurons from iPS cells are also a good platform to test or screen a high number of compounds with potential therapeutical efficacy. In this study, for example, candidate drugs were first assayed in these human neurons in vitro, and where the electrophysiological and morphological features were recovered by some of them. And then the good candidates were then tested in vivo in animals, also showing some positive results. The test of potential therapeutic agents can be sometimes a consequence of the molecular data and studies that iPS cell-derived neurons can provide. In this study from Park Lab, an unknown aspect of MECP2 function when regulating other genes was uncovered, and this inspired the use of a particular compound, which then showed therapeutic potential, since it can restore certain neuronal defects. For example, here we showed how calcium signaling is recovered, so that now the cells treated resemble more the wild-type cells. Also, very recently, the Finnish lab has used similar to the two-dimensional models to assay a strategy to recover MECP2 function based on the manipulation of certain DNA modifications, certain features, and certain factors that bind to chromatin to reactivate the X chromosome that contains the normal MECP2 version of the gene. So, here in these images, you can, as an example of this study or a readout of this study, you can see how the red color in the control cells or the control neurons is absent in the red neurons. This color detects MECP2, the normal MECP2. And when the genetic intervention is implemented, we recover the expression of MECP2 in these neurons. So while it is true that 2D models represent highly reproducible cultures with homogeneous distribution of nutrients and small molecules and are very handy for mechanistic studies and drug testing, 3D models allow the proper formation of distinct cell layers, synaptic connections, and networks that across a specialized neural cell type, so that 3D organoids allow a closer mimic to the in vivo steps of human fetal neurodevelopment and maturation. So let me mention a few works dealing with red organoid generation. This first study from Finnish lab, for example, addressed molecular aspects of MECP2 function and link them to structural defects observed in neurogenesis by analyzing different organoid zones. For example, they observed more expanded ventricular areas. As mentioned before, the organoid field is quickly evolving, and different labs have reported the methods to fuse organoids from different brain regions. For example, dorsal and ventral forebrain organoids can be fused in vitro, and their links and their interconnections can be studied. This was applied in a Rett study recently, where ventral and dorsal specific or related organoids were separately generated and then fused in vitro, and the migration of neurons from the ventral zone to the dorsal zone was then tracked by labeling the ventral neurons in green. And here we can see how in the control situation or the normal situation, these neurons appear more go to the dorsal zone, whereas the red neurons show defects in that regard. Lastly, I'm showing now some results from a work by Alison Moatry's lab, which I think illustrates very well the power of combining 2D and 3D models. In this case, a battery of candidate compounds were tested in 2D neurons to then assay them, the ones that showed some effect, assay them in more complex 3D organoids and with a more in-depth characterization of their therapeutical potential. With all this, the main take-home message I would like to convey to you today is that human neural models, both 2D and 3D systems, which are getting increasingly more complex and sophisticated as they incorporate even non-neural, non-central nervous system derived entities such as blood vessels or microglia, are excellent platforms to recapitulate and to characterize Rett developmental features. And they also allow drug discovery and the analysis of drug responses. Thank you very much for your attention. I'm leaving here my email in case you wish to contact me in the future for any questions. Thank you.